Alright, our element is arsenic, represented by As on the periodic table. It's a steel gray metalloid with an atomic number of 33. It comes in 3 different colors, but the most stable of the 3 is steel gray. There are 33 electrons and 42 neutrons in the most stable nuclide. Arsenic is notoriously poisonous, and when it's burned, it forms a very toxic white cloud of arsenic dioxide.
Alright, here's an example right here, arsenic. Alright, arsenic is important to our studies of environmental science for a number of reasons. First, arsenic is a byproduct of refining the ores of other metals, such as copper and lead. More importantly, the majority of the arsenic released into the atmosphere is due to the burning of fossil fuels, nearly 80,000 tons per year. Arsenic is mainly emitted by copper-producing industries, but is also produced during lead and zinc production and in agriculture. It can be destroyed once released. Once released into the environment, and causes health effects to humans and animals. Some long-term health effects on humans due to exposure to arsenic include increased chances of cancer development, infertility, and miscarriages with women, heart disruptions, brain damage, and damage to DNA.
I'm Michael Malahy.
I'm Amy McCosker.
Okay, so for our project, we're trying to figure out the factors that affect the rate of oxygen production in photosynthesis.
Alright, for our project we're using a GLX device, and we have it hooked up to a thermometer and an oxygen tester. And these things are in a cup of water, and we're testing how much dissolved oxygen is in this water and the temperature. And here are our findings.
Photosynthesis is the process in which plants take in energy from the sun to produce glucose. Glucose is a fundamental energy source for living organisms. In order for this to happen, sunlight, carbon dioxide, and water must be present.
We predict that the heat in both the non-plant and plant water will increase because the lamp provides a heat source and it is absorbed in the water. But we believe that the water without the plant, the oxygen level will stay the same, whereas the water with the plant, the oxygen will increase because of photosynthesis.
Okay, as you can see from our graphs, the results of the experiment are the temperature in the first. As you can see from our graphs, the temperature in the first experiment without the plants in it started at 23.5 and gradually increased to about 24. And our second experiment with the plants inside the container started at about 24.5 and ended about 25.5, and that was the temperature. And the oxygen level in the first experiment without the plants stayed the same throughout the whole thing at about 6, and the oxygen level in the second experiment with the plants inside the container has gradually decreased.
We believe that the temperature went up because the lamp provided a heat source that was absorbed by the water, and we believe that the oxygen level stayed the same with the non-plant because there was no change. But when the plant was added to the water, we believe that the plant needed a supply of oxygen, so it absorbed the oxygen from the water.
So if we could start or do a new experiment, we would have the same starting temperature of the water from when from the plants weren't in and from the plants were in. As you can see from the graph, there was a big difference here, and if it was the same, it might have been a different results than what we got. And also, we would try to use multiple different types of plants that weren't all the way underwater, and see if that would affect the oxygen rate.
We conclude that photosynthesis most likely did not occur during the time period that we recorded. Although we thought at the beginning that the plant would actually produce more oxygen, it ended up taking away oxygen. So now we understand that it most likely takes longer for a plant to produce more oxygen.
I'm John Ford.
I'm Kyle Dennis.
And I'm Worthy Patterson, and this is our experiment.
Right now we have our control test going. We have two sensors in this inner cup of water.
The first sensor controls temperature, and the second controls oxygen.
You'll see that down there. We have this hooked up to our GLX, which is then hooked up to a computer. As you can see, our control is pretty flat there. And we have a light going to represent the light that we'll have when we later put a plant in here to read the change in oxygen levels during photosynthesis.
The purpose of this experiment is to see the change in temperature and oxygen levels of photosynthesis in water.
Photosynthesis. Photosynthesis is how a plant makes its own food by using sunlight, carbon dioxide, and water to produce oxygen and glucose. There are two main parts to this reaction. There's the light reaction and the dark reaction. The dark reaction is also known as the Calvin cycle. The light reaction is when the plant takes energy from the sun that is absorbed by the chlorophyll molecule in the chloroplast of the cells, and the ATP molecules are produced. During the dark reaction, or Calvin cycle, the ATP molecules release their energy to form glucose molecules from the carbon dioxide and water that enter the leaf through the stomata.
We predict that the amount of dissolved oxygen will increase because of the oxygen given off by the plant during photosynthesis. The temperature of the water, however, will remain constant, or at least only increase a little bit because it will not have a great effect on the temperature.
These are the results of our experiment.
Okay, this orange line right here, as you can see, is the temperature. In the first experiment, it was very low. In this next experiment, it went a lot higher. This could have been due to the light focused on it. The light had been focused on it for longer, so the temperature could have risen. The line is very steady, so the light seems like the best reason for the increase in temperature. The oxygen increased throughout the experiment as shown by this blue line. This red line is the first experiment in which it had very little increase. And not only is the oxygen higher from the beginning, but it increases throughout the experiment. This is possibly because of the plant going through photosynthesis as the experiment went on.
We believe that a better way to have done an experiment would be one very similar to this, but instead of using a light like this, where it is artificial and turned on and off at will, if it had been done perhaps in the sun, where we have a constant heat, so we have no changing variable of for the heat for the water, so the water would be at its maximum heat before the experiment was started.
Our hypothesis was true, but not by some of the reasons we expected.
For example, the rise in temperature throughout the experiment may have been caused by this light, which was closely focused on here. So it may not have been something that the plant was actually doing to the water, but rather the fact that there was heat concentrated so close to the water.